deals with synaptic plasticity and how the strengths of synapses can be changed. And this is incredibly important. And it's important because, I don't know, for years and years and years, I used to think of the brain as being a hardwired system. I mean, the wiring was very complicated, but it was basically hardwired. If one thing was connected with another, it was connected. It turns out the connections are incredibly plastic. They can be strengthened, they can be weakened, they can be eliminated, they can be made. I mean, it's really quite extraordinary the way the brain can construct itself. And this little figure down here simply shows, an action potential coming down. And I'm going to deal with two new types of receptors. The action potential, calcium comes in, synaptic transmitter is released, and it's released and then bound onto these receptors. And we're going to talk about two of these receptors today. They're both glutamate receptors because glutamate is the main receptor, the main excitatory neurotransmitter in the brain, and they are called AMPA and NMDA types of receptors. And the third thing is we're going to consider the role of the NMDA receptors, in particular, in regulating calcium influx into the postsynaptic cell. Right? There it goes. Look, if you watch it down here, you can see all the action. And I'm going to consider how calcium influx can activate enzymes and how those enzymes can strengthen a synapse by adding new receptors. Watch, look at that. Amazing. And then how it can weaken a synapse by removing the receptors. And the fourth thing I'm going to discuss is why these features might underlie learning and memory. Okay. So with that introduction in mind, we're first going to discuss the two types of glutamate receptors, the NMDA and the AMPA type receptors. This is just to remind you that glutamate is the major excitatory transmitter in the brain and spinal cord. And the way these things work are as follows. So this is the AMPA type of glutamate receptor. This works just like the acetylcholine receptors I described when we were talking about the neuromuscular junction. So there is a receptor up here, but in this case it binds glutamate, and there's a gate, and when the gate opens, there's an ionotropic channel. There's a channel associated, attached to the receptor, part of the receptor, and current will flow. And the reason it's called AMPA is because this particular chemical, this is it, alpha amino 3 hydroxy 5 methyl, blah, 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 blah. That's, everybody just calls it AMPA. That can act as an agonist, that is a substitute for glutamate to open the channel. But normally glutamate is released, and the glutamate will bind on it, will open the channel, and the channel then becomes equally permeable to sodium and potassium. And it drives the membrane potential to the reversal potential, which is about 0 millivolts. So it's very, very depolarized. And the second channel is the NMDA channel. And this one I can remember. It's N-methyl diaspartate, but nobody ever calls it that. Everyone only calls it the NMDA receptor. And the idea, again, is NMDA can act as an agonist for glycine right here, the glycine, for glutamate right here. But normally glutamate is released, and when it binds onto the receptor, it opens the channel. It's like any other channel, except this channel is plugged. It has a plug of magnesium ion in it, doubly positively charged, and that prevents any ions from moving through the channel, even though the gate is open. So the way this works is it works together in tandem with the NMDA receptor, with the AMPA receptors. So the AMPA binds the glutamate, it opens up, and it drives the membrane potential right around here to the reversal potential, which is very positive, It's about 0 millivolts, at least in this little region, it's about 0 millivolts. And that in turn will expel the magnesium ion and allow current to flow through the NMDA receptor. And the important point about this is that the current that flows through it is calcium. And remember, whenever calcium comes into the cell, all hell breaks loose. Right? There are all kinds of consequences for it. And that's going to be the subject we're going to deal with. And before I tell you exactly what those consequences are, I want to introduce you to this gentleman here. Uh, his name is Donald. His middle initial is O, and his last name is Hebb, Hebb. He is extremely famous. He is a Canadian psychologist whose book was published in 1948, and it had profound influences on our understanding of the neural basis of learning and memory. In fact, he was really the first major person to discuss what the neural basis of learning and memory might be. And his book is called The Organization of Behavior. It's extraordinarily well known. You've got to know who Donald Hebb is. And here's what he proposed in that book. He proposed the nervous system learns by synaptic strengthening. And he had two types he spoke about. One type is called specific strengthening. By that he means, what is the strengthening that occurs between one individual axon here and its postsynaptic target, which here shown here in red. And what he wrote is, when an axon of cell A, right here, this one right here, is near enough to excite cell B, the red one, and repeatedly or persistently takes part in firing it, some growth process or metabolic change takes place in one or both cells, such that A's efficiency, the efficiency of this synapse, as one of the cells firing B, is increased. And just to give you an idea of it, this is what it is. High rate of firing, and all of a sudden the synapse is strengthened. So the next time this fires once, bingo, you get a much bigger response in the postsynaptic cell. And that's what he meant by specific strengthening, the strengthening of one synapse onto its postsynaptic target. But he also proposed something that we'll talk about a little later. This we're going to talk about immediately in just a moment. This we'll talk about a little later, and it's called associative strengthening. And what he said is that groups of neurons, which tend to fire together, form a cell assembly whose activity can persist after the triggering event and serves to represent it. I will decipher that for you and translate that for you. And it says this: cells that fire together wire together. Right? And it's a famous saying, and it refers to the idea of associative strengthening. That refers to the activity of groups of neurons and how they might strengthen synapses together. I will come back to this in a little while. First, I want to take up the idea of specific strengthening. And the idea is going to be that high rates of activity can strengthen synapses through a process called long-term potentiation, or LTP. This is a standard term now in all of neuroscience. You've got to know what it means. And here it is. I'm going to illustrate it. Here's a neuron, and it's very active. It's firing a lot of action potentials, making a synapse to a postsynaptic cell. And this one happens to be inactive. It's not firing at all. All right, I'm going to draw them. So this is the postsynaptic cell, my drawing of it. This is one cell, pathway 1, it's in red. This is another cell 2, pathway 2 is in green. And I'm going to record from this cell intracellularly. I'm going to stick an electrode in it. And what LTP, or long-term potentiation, it's the strengthening of one synapse due to high rates of activity in that synapse. And the one we're going to demonstrate it for is for cell 1 and this pathway. And here's the way I'm going to do it. I'm going to put stimulating electrodes on cell 1 and stimulating electrodes on cell 2. And then I'm going to stimulate cell 1 with a single pulse of electricity. Watch, bingo. One pulse, one action potential, and I'm going to look at the postsynaptic response. Here it is. And then I'm going to do the same thing to cell 2, or pathway 2. A single action potential, boom, boom, a single postsynaptic potential. In other words, it released a little bit of chemical, and it caused the membrane potential here to change by 5 millivolts, 4 or 5 millivolts. And it did the same if I stimulated this particular pathway. All right. But now what I'm going to do is something a little different. I'm going to stimulate pathway 1 strongly, titanically. What titanically means, I'm going to stimulate it at a rate, at the highest rate it's capable of firing. It can't fire any faster. So I'm going to stimulate it really strong so it fires like mad. Watch. All right? And then I'm going to stop. And a moment later, I'm going to come back and I'm going to stimulate it with a single stimulus. Bing, watch. Bing, bing. And now the response is double or more than double the size than it was before. This synapse has become strengthened. Right? Something has changed. And it's not true in synapse 2 because synapse 2 has not, or pathway 2, has not been titanically stimulated. If I stimulate it with a single shock, nothing happens. It's the same as it was before. Right? Only specifically pathway 1 was strengthened because only synapse 1 was strengthened because only synapse 1 was titanically stimulated. All right? Okay, are we okay so far? Any questions so far? We all right? Okay. So I want to show it to you over time. And just to give you an illustration, the time period here is in minutes, not seconds, not milliseconds, minutes. So I'm going to stimulate both of these, boom, 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 and I get responses. And then what I'm going to do is I'm going to stimulate 1 titanically. And then I'm going to wait a minute and I'm going to stimulate it again. And look, the size of the response is much larger. But the point of this is that amplitude increase, that enhancement, that potentiation of the postsynaptic response lasts for more than an hour. Hence the term long-term potentiation. And if I stimulate pathway 2 with a single pulse, boom, 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 what happens is nothing. It has not changed at all. Only pathway 1 has been potentiated because it was stimulated titanically here. That's the idea. Okay. Let's go on. What I want to show you here is that both of these synapses have both NMDA receptors and AMPA receptors. So synapse 1, when glutamate is released, it will bind onto both AMPA and NMDA receptors. And the same with synapse 2. It has AMPA and NMDA receptors as well. So what causes a high rate of activity in synapse 1 to generate long-term potentiation? And the answer is that a large calcium signal triggered by high rates of firing in the presynaptic neuron activates an enzyme, a protein kinase, that adds phosphate groups to other proteins, which triggers a series of events that strengthens the synapse. And I'm going to show it to you diagrammatically. And here's going to be the idea. The idea is going to be that when this cell, this presynaptic cell, is stimulated very, very, very strongly, so it releases a lot of glutamate, what's going to happen is it's going to bind onto the AMPA receptor, it's going to expel the magnesium, and there's going to be a large influx of calcium into the into the postsynaptic terminal. This is the postsynaptic terminal. This calcium, in turn, in ways that I will show you in just a moment, is going to bind on to a calcium binding protein called calmodulin. And if you remember, the same thing happens in the presynaptic cell. When calcium comes in, there is calmodulin, it binds onto the calmodulin, and the activated calmodulin activates a kinase which phosphorylates things, which causes the release of the vesicles from the cytoskeleton, the actin cytoskeleton. In the postsynaptic cell, there's also calmodulin, same type of calmodulin. It is activated by calcium, and it activates a protein kinase, a different protein kinase. This protein kinase is called protein kinase 2, and it does 2 things. One, it will add, well, it will activate a whole series of events. And I want you to appreciate that I'm not going to ask you to learn all of these different events, but it activates a series of events which ultimately will cause the insertion of extra AMPA receptors into the postsynaptic cell, like that. So now, if glutamate is released, it doesn't have just one AMPA channel to go through, it has three AMPA channels to go through. And of course, you can bring through a lot more positive charge and make the depolarization larger. In addition to that, the kinase will also phosphorylate the AMPA channels. And what the phosphorylation does, it's not so obvious in this picture, but it makes the conductance, the channel size here, larger. And that will allow more current to flow through the channel. So not only do you have more channels, each channel is more conductive. So you're getting a lot more positive charge in here once you release a glutamate onto the synapse, right? And those are the two things that generate long-term potentiation. Now, let me stop. Is that okay so far? Does anyone have any questions about that? Sure.
Dr. Pollock, we have a question from Derek in the chat. Derek, feel free to unmute if you'd like to ask. I'm also happy to read your question.
Go ahead.
Okay, awesome. So looks like Derek was asking, when tetanus is applied, we see that tetanus has a similar graph to whenever we apply Essarin to that graph from the previous lecture, right? So Derek's question was that tetanus is inhibitory, right? So shouldn't it hyperpolarize instead of depolarize?
No, tetanus simply refers to the fact that the cell is firing at its tetanic potential. It's firing as fast as it can. You make the cell fire tetanically. There's nothing with excitation or division. You're just making that presynaptic cell fire as fast as it can. That's all it means. I don't understand where the negative comes from, and presynaptic and all that other stuff. What do you mean? All it means is you're gonna put an electrode onto the presynaptic cell, onto this cell right here, this one, and you're gonna make it fire by electrically stimulating it, and you're gonna make it fire as fast as it possibly can. Boom, 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 boom. That's it. That's all it means. And if it fires fast, it's going to release a lot of transmitter. And if you're asking, do these vesicles have to be recycled? Yeah, they have to be recycled.
Okay, I'm sorry. Some confusion against, like people were thinking about like the toxin itself versus just the term tetanically, right? So, thanks for clarifying that. We had one more question in terms of why do more AMPA channels appear again?
Why do you want AMPA channels to be there again?
Oh, why do more AMPA channels appear? Because it says added to the.
Why are more AMPA channels inserted into the membrane when you do this?
That's the question. Yes.
Yeah. I don't know. Ask God the next time I speak to him. I don't know. All I know is by adding more AMPA channels, you're going to make the synapse stronger, and that's what you're trying to do. I don't have a better answer than that. I don't have a better answer. I emphasize the idea that there are two things going on. I am going to explain the phenomenology to you, and maybe on a certain level the mechanism. The question of why is an entirely different question, and the whys of these things I know very little about. I don't know if anybody knows about. The net effect, though, of putting more AMPA channels in is that you make the synapse stronger because you're opening up more channels and more current can come into the postsynaptic cell. I'm sorry, that's about the best I can do. Is that okay? Yeah. Okay.
The students are great. Thank you.
Okay, let's go on. So I want to come back to the idea of the Cal module. There it is. Remember, this is not the presynaptic, this is the postsynaptic cell. There's the calmodulin, the little mouth here. The little mouth is going to, that's my drawing. It's going to bind the calcium. When it binds the calcium, it's going to activate, right? And when it activates, it will activate the protein kinase. This particular protein kinase is protein kinase 2. And it's going to phosphorylate lots of other proteins. And one of the things that it's going to phosphorylate are the AMPA channels. And it's going to add a phosphate group onto them. And when it does, look, the conductance is going to make bigger. The channel is going to get bigger. This is a normal AMPA channel. This is one that's phosphorylated. They're going to open them with glutamate. Watch. And you're going to get a certain amount of current through here. And you're going to get a larger amount of current through here because the channel's bigger. And that's one of the ways in which the synapse is made stronger. Because the more positive charge you come in, the more depolarization you get in the postsynaptic cell. And in addition to that, it triggers a series of cascades. And those cascades, the ultimate effect is to add AMPA channels onto the postsynaptic cell. So you not only have more AMPA channels, they're phosphorylated. And that's what makes the synapse so much stronger. And the kinase activated by calmodulin is called a calcium calmodulin dependent protein kinase 2, or CAM kinase 2. That's what it's called. Right? Okay. So there was another important word on there that I didn't mention, and I certainly didn't play upon, but I'm going to mention it now. And that word is persistent. And what it means is that if you have a small calcium signal, a little bit of calcium comes in, there will be a transient amount of kinase activity because some of the calcium binding onto the calmodulin will activate a few calcium CAM kinase 2s, and as soon as the calcium is sequestered, the effect is over. That's it. It's called transient. If you get a large calcium signal, something else happens, something very important. The calcium binds to the calmodulin binds onto the CAM kinase 2 to a much larger degree, and it causes the CAM kinase 2 to have a persistent effect. And what that means is even after the calcium is sequestered, the CAM kinase 2 activity, its enzymatic activity continues. It's persistently activated, and it thus can produce its effects even in the absence of calcium, at least for a period of time. And let me try and illustrate this to you. Here we have calmodulin. Here we have an inactive kinase, and we're going to get some calcium, a large calcium. It's going to activate a lot of the calcium-activated calmodulins, which are going to activate the CAM kinase 2 to a very large degree. And what's going to happen is the large degree of activation is going to cause, in ways that I will describe to you in just a moment, the CAM kinase 2 to autophosphorylate. It phosphorylates itself. And what that means is even after the calcium calmodulin levels drop off and the calcium is sequestered, this enzyme remains active even in the absence of calcium because it autophosphorylates. And it can exert its effects, and that's extremely important. Now I want to consider the structural features of CAM kinase 2 and how those features can explain why there's a weak CAM kinase 2 response to small calcium signals and why large calcium signals generate a persistently active kinase. So I just want to point out to you that each molecule of CAM kinase 2 is composed of 12 subunits. This is the subunit. And each subunit is composed of two portions. The red portion here, which is called the autoinhibitory portion, and a green portion here, which is the catalytic portion. And normally they're joined, and the catalytic portion is inactivated by the autoinhibitory portion. And now, this is just one subunit. There are 12 subunits normally on a CAM kinase 2 molecule, and they're organized as the spokes on a wheel. Right? And here's going to be the important part. This is the catalytic unit. I don't care about the residues. I'm not going to ask you about the names of the residues. What I want you to know is that right here, this is the catalytic domain, or the catalytic component of this catalytic domain. And this autoinhibitory domain right here, this holds this together. And when these are held together, the catalytic domain is inactive. The third thing I want to point out to you is right here. This is where the unit or the subunit can become phosphorylated automatically. It can become autophosphorylated. Let's put it that way. So let me show you how this works. I don't care, you can call this pseudo subclavier. This little region is the region where the calcium calmodulin, the activated calcium calmodulin molecule, binds to. And when it binds to this subregion, what it does is it causes these two to separate. Yeah, that's the part that can be autophosphorylated. All right, that's not what I want to show you. Come on. Disassociate. That is the part that can actually do the phosphorylation. And, yeah, I'm not explaining this well. Let's go through this. I thought I had this on this slide. Here it is. Here's the calcium activated calmodulin. It's going to open it. And when it opens it, this becomes active. This can now phosphorylate a substrate. Right? And this is the way they're arranged on a spoke. There's only half of them. Right? But the point is, if you have a large calcium signal, all the calcium calmodulin are all here. And they've opened up all of the subunits. And if the subunits are open, what it means is that this part, which can phosphorylate things, can also phosphorylate this. Autophosphorylate. And this one can autophosphorylate this, and this one can and that's the way these become autophosphorylated. So this thing is operating to not only phosphorylate itself, but to phosphorylate other proteins. And the point is that when it becomes autophosphorylated, even in the absence of calcium, when these calcium calmodulin molecules disappear, these things remain open, and these portions can phosphorylate a substrate. That's the idea. That's why autophosphorylation is so important. So the idea then being, if you have very little calcium, the chances of the calcium activating calmodulin binding to adjacent or neighboring subunits is very small. And if the neighboring subunit isn't open, this cannot autophosphorylate this. It's got to be open. And this cannot autophosphorylate this because this one isn't open. They must be open. And that's why a large calcium signal opens a number of them and opens neighbors that allow them to be autophosphorylated. Can't do it. So as soon as the calcium is sequestered and the system is over, bingo, dingo, the effects are over. That's why it operates very transiently with small calcium signals. Okay. I want to make one other point. Let's assume we had a fairly large calcium signal and we autophosphorylated 3 of the subunits. Well, if another calcium signal comes along before these things can be dephosphorylated, voila, they can cause the autophosphorylation of even more units. And this thing can become even stronger than it was before. That's the point. This is not either or. This can be done sequentially to increase the efficacy and the strength of the phosphorylating ability of the CAM kinase 2. Okay, let me pause for a moment. Does anyone have any questions about this?
Yeah, there's a brief clarifying question about where on the subunit does CAMK2 get autophosphorylated?
Right here. I forget the number. It's right here. That's where it gets that autophosphorylated. Right here. And that's the idea. And when it does, it causes the the, in other words, it autophosphorylates the inhibitory subunit right here. I forget the number. I can look it up. We can go 2 slides back. When it does, it causes the inhibitory to separate from the phosphorylating subunit, and it's able to phosphorylate anything. Then it becomes active if it's separated. Does that answer your question?
Apsaline says yes, thank you.
Yes. This is, hold on, 319, 310. Ah, 286. All right. That's the site. We're on 286. But this, for our purposes, the number isn't important. The point is, if it is autophosphorylated by its neighbor, then in fact it remains open so long as it's autophosphorylated. That's the point. And here you can see this is transient, no autophosphorylation. As soon as the calcium is not being secreted and it is sequestered back into mitochondria or pumped out of the cell, the effect is over. On the other hand, if you have a large calcium signal right here, the effects are persistent because it autophosphorylates itself, and that's shown up here. The autophosphorylation of some of the subunits. And now, if you have a second calcium signal that autophosphorylates even more of the subunits, you get even a larger response. That is, the kinase activity becomes even stronger because they add. So this is a very, very powerful kind of thing. This is just a review. A large calcium signal will basically cause the CAM kinase 2 to become persistently activated. It causes a whole series of reactions. The net effect is to insert more AMPA channels into the system and also to phosphorylate. So there's more current, and then they become phosphorylated so that they can actually allow even more current to come through each of the channels. And hence, the synapse is strengthened. And they can be even strengthened further by another large signal coming in that would cause the persistent inhibition to continue, but to be even stronger. Okay. So, what I've shown you here is specific strengthening, how the activity, high activity in one neuron can strengthen the synapse. It strengthens it postsynaptically by inserting additional AMPA channels into the synapse. It does not affect a pathway that is inactive. So, I want to introduce you again to Donald Hebb. And Donald Hebb now had a very, very important idea, and it's known as Hebb's postulate. And that could account for the cellular basis of learning. So an important aspect of Hebb's postulate is that a given presynaptic input to a cell need not by itself be of sufficient strength to induce a large depolarization in its target. And you're saying, wait a minute, that's not what you just told me. Well, you just told me it has to be. Well, what he's saying, let me finish. He's saying it doesn't have to be. If, however, that input fires at the same time as a number of other inputs, and their combined action strongly depolarizes the cell, all of these inputs will tend to be strengthened. Thus, two or more inputs that fire at the same time become strengthened. In short, inputs that fire together, wire together. Right? And let me show you how this works. It's not that difficult. And just as an illustration, I want to just show you classical conditioning that was first introduced by this very distinguished gentleman over here, Ivan Pavlov. And the idea was Ivan took a dog and he presented some meat to the dog. It wasn't a Big Bell hamburger, it didn't have Big Bell in those days, but you get the idea. And the dog salivated. And then what he did is he rang a bell. And when he rang the bell, the dog didn't salivate. Why should he? Until he paired the meat with the bell. And the dog salivated. He was salivating, obviously, to the meat, but he was now learning to associate the meat with the bell. And then when he heard the bell alone, he thought he was going to get some meat, and he salivated to the bell alone. Right? So the bell becomes associated with the food. So if you want to, this is the high rate of activity in this circuit. This is a low rate of activity, which doesn't itself, add any synapses, doesn't strengthen them. But when you add the two together, you strengthen this one. Inputs that fire together, wires together. Now, how does this work? So here I have two systems. One system here, a lot of activity that will strengthen by long-term potentiation. This one, it will have no effect at all because it's not firing. Here, I have a lot of activity that will strengthen, and here I have weak activity. But if this strong activity can come down here and affect this postsynaptic cell here, I can strengthen this fire as long as they fire together. And let me illustrate this to you. And I have to illustrate this to you now with a little bit more of a realistic situation. This is the typical, neuron in the cortex or the hippocampus or someplace like that. And if you notice, here's the cell body, and it's got a axon coming out right here, this black one, and it's got numerous dendrites. Look at all the dendrites it's got. And if you notice, the dendrites got these little bumps on them. These bumps or small protuberances are called spines. And let me illustrate what spines are. Now, let me just make one other point. Not all neurons have spines. Here's another one. Doesn't have spines, right? Only this one does. A lot of neurons have them in the hippocampus cortex, but not every neuron does. Right, so here's spines. I'm going to blow this up. This dendrite right here with all these spines, and I'm going to blow it up. There it is. And here are all the spines. You can see them. I'm going to label them. Look at them. They're all over the place. Now, here's the point. I am going to take two spines. I'm going to draw them. This is my drawing of two spines. Right? And here they actually are in electron micrographs of some of these neurons, right here. And I'm going to draw them. This is hypothetical. Here's a spine. Here's a spine. And it turns out all the spines have both AMPA and NMDA receptors. And they all are excitatory synapses. They all receive glutamate from their presynaptic targets. These are not inhibitory synapses. Spines are not inhibitory. They're excitatory. Okay. Now, let's take two spines right here. There's one spine, and here's two spines, one right next to the other, just for the hell of it. And here's an axon terminal, here's another axon terminal. There are the AMPA receptors, 1, 2. Okay, here are the NMDA receptors, and this little black thing in the middle is the magnesium plug. Right, and let's see. Oh, here it is. And look, there's one spine, and there's the other spine. That's my drawing. Okay, and now we're going to look and see how the activity in one spine, high activity, can strengthen the synapse in another spine that has low activity. That would not normally strengthen it if that low activity occurred by itself. But if that low activity occurs in the presence of high activity in spine 1, it will strengthen spine 2. And here's how it works. Here's the low activity in spine 1. Low activity and high activity in spine 2. So the high activity in spine 2 is going to strengthen the synapse in spine 1, which would not be strengthened by this low activity. But if they occur together, they will. Neurons that fire together, wire together. Now, okay, now, here they are, my drawing. Again, AMPA, NMDA, NMDA, AMPA, okay, magnesium, magnesium. Probably going to show you. There's the NMDA. There's the magnesium. The pores in the NMDA are blocked by magnesium. Yes, there's the spine. Here are the reserve AMPA channels. There are always reserve AMPA channels in neurons. So what we're going to do is we're going to stimulate axon 1 weekly. There it goes. And then we're going to release some glutamate, and that glutamate is going to open up just a few of the AMPA receptors, and you're going to get a very weak depolarization, maybe 5 millivolts. It's not enough to expel the magnesium ions. So all you do here is you're not strengthening this. There's no long-term potentiation going on here, and you've got a small, very weak excitatory postsynaptic potential because of the influx of a little bit of positive charges carried by sodium. Okay. Okay. All right. Next, I'm going to cause axon 2 to fire really strongly. And it's going to release a lot of glutamate. It's going to bind that. It's going to bring in a lot of positive charge. It's going to expel the magnesium ions. And it is going to basically bring in calcium and cause, if you will, the phosphorylation and the addition of calcium of AMPA channels onto the synapse. So synapse 2 will undergo strengthening. It will undergo long-term potentiation. And here's the response to it. It goes from -70, -45, 35 millivolts. But this one didn't fire. So nothing happened here. Nothing. Now, we're going to fire both of them. This one weakly, this one strongly. But they're going to fire at the same time. A lot of it. A little bit, a lot. Right? Expel the magnesium. Right? And this big depolarization with the calcium, this big depolarization is going to spread because this is more positive than this, much more. So the positivity is going to be attracted. And if this is close enough, you're going to get positive in here. And those positive charges can then expel the magnesium, causing the influx of calcium, causing a little bit of long-term potentiation. Right? You can insert one more in here. You might phosphorylate them. Here, you're not going to insert one. You might insert 2 or 3. So the strengthening of the synapse is going to be much larger than the strengthening of this one. But they're both going to be strengthened, even though the activity in axon 1 by itself was not sufficient to cause LTP. It was only sufficient to cause LTP if it's in the presence of high rates of activity in axon 2. Cells that fire together, wire together. Okay? Does everyone see the logic of that? So, now I'm not going to show you this. I'm not going to go discuss this with I will. So what you can have, and this happens particularly during development, you have what are called silent synapses. They won't fire because they only contain NMDA receptors, only. So if you fire this and you cause a release of glutamate, nothing happens because it opens the channel, but there's not enough positivity in here. There's no positivity to expel the magnesium. So nothing happens. It's silent. No response. Then what you do is you fire this like crazy, and you have the same firing rate here that usually would do nothing. But this, of course, expels magnesium, brings in this. You have a large depolarization. That depolarization can then spread. And it can spread and expel the magnesium ions from the NMDA receptors, allowing calcium to come in. A little bit. Come on. There it is. Causing a few AMPA receptors to be inserted into the channel. And the channel, which previously was silenced, is no longer silenced because it has been strengthened by the charge that has moved from here to here. Cells that fire together, wire together. Right? This is known to happen. So if LTP, let me stop. Is that okay? Does everyone understand that? Okay. If LTP is the yin of synaptic plasticity, LTD, long term depression, is the yang of synaptic plasticity. Okay, so here it is. What long term depression does is exactly the opposite of long term potentiation. Instead of being evoked by high rates of stimulation, it is evoked by very, very low rates of stimulation for fairly long periods of time. So if I have stimulating electrode, right, and I fire it, I go single electrons. I get my control response, bingo, right there. And then what I do, there it is. And then what I do is I stimulate this at very, very low rates for quite a while. I go bing, bing, bing. And I keep doing this for, I don't know, 20 minutes, half hour, something like that, 15 minutes, whatever the case may be. And then I stop and I come back and I stimulate this with a single pulse. Ping! Look what happens. Now the response is smaller. The synapse has been weakened due to low rates of activity in the presynaptic axon. And you might say, well, how does this work? Well, I can look at it over time. I was stimulating here. And now I stimulate very, 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 very slowly here. And bingo, I come back and voila, what I got is a reduced. And that reduction is known as long-term depression. And you might be saying, well, how the hell does that work? Well, here is the idea. The idea here is going to be that you have calmodulin. And what the calmodulin can do is it can bind calcium and become activated. And when you have high rates of activity, you, of course, will activate the CAM kinase 2, calmodulin CAM kinase 2, which in turn will phosphorylate other proteins. And if it's a high rate of activity, it will be persistent. It is very important that it's persistent. On the other hand, the calmodulin that binds to CAM also activates a phosphatase, something that removes phosphorus groups. And that phosphatase is called calcineurin. And calcineurin is also activated by calcium activated calmodulin. Woo! We got two things going on that are competing with each other. We have the CAM kinase 2, which phosphorylates things, and we have the calcineurin, which is a dephosphorylating agent. How does this work? Well, here's the idea. The idea is going to be that you have a synapse here, right, here, and this is called the postsynaptic density. And it stains density, and it's got all kinds of stuff in there. You know, it's got calmodulin and kinases and phosphatases, and it's got everything. It's a lot of stuff going on here. Okay. And here's the idea. With LTD, what you've got is you've got a low rate of activity. That is, you get calcium in, but very little. And that calcium then activates a phosphatase, and the phosphatase will do two things. It will sequester the AMPA receptors, and it will dephosphorylate the AMPA channels that are inserted in the membrane. So now you've got exactly the opposite of what you had with high rates of activity. How does this work? And this is my understanding. So here, we're going to have a small rate of activity, transient. So you're going to have some calcium come in, it's going to activate some calmodulin, and that calmodulin is going to do two things. It is going to activate the calcineurin, and it is going to activate the CAM kinase is activated weakly, right, because only a few of the subunits are activated and there is no persistence. This is stronger. Not that much stronger, but it's stronger. So the net effect of each small influx of calcium is to have the calcineurin dominate. And if you do this again and again and again and again and again and again and again, the calcineurin will ultimately overcome all the effects of the of the calcium activated calmodulator, the CAM kinase 2. It will win. It will win. And that's the idea. That's why you have to have low rates of activity for quite a while for the calcineurin to win. Because every time it's activated, it has a little bit more activity than this, but then it stops. And now you get another signal, a little more activity, stops. Another signal, a little more activity, stops, stops, stops. And you get the idea. Okay? So calcium, CAM kinase 2 and calcium are only activated as long as the calcium is in the cytoplasm. Now we have a large calcium signal. And it's going to activate this to a very large degree. And it's going to activate this to a very large degree because a lot of these are going to become autophosphorylated. Now, who's going to win here? I don't know. I'm not really sure. But calcium then is going to go away. But, and this is the but, this is no longer active. No more calcium around. It's gone. But this is because this is going to be autophosphorylated. And as a consequence, it is going to exert its effects, right? Even in the absence of calcium. And in the absence of calcium, this is inactivated. There's no effect. So this can go on exerting its effects without having to compete with the calcineurin because it's autophosphorylated and persistently active. Persistently, right? So calcineurin is only activated so long as the calcium is the cytoplasm, but CAM kinase 2 is persistently activated. And the persistent activity of the CAM kinase 2 allows it to activate cascades without competition from calcineurin. And that's why high rates of calcium, which autophosphorylate the CAM kinase 2, win out and produce long-term potentiation. This is it right here. Right here. And now comes the interesting part. The interesting part is that these two can act in opposition to each other. In other words, you can produce CAM kinase 2 right here, right? And you got big response. And now you just start going slowly, 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 slowly. And what you can do is you can reduce the effects. So you actually get, after you've gotten high frequency stimulation with potentiation, you can completely eliminate it and produce depression with very low frequency stimulation. These two are in opposition to each other. You can, of course, then take something that has been low and produce high if you then stimulate it strongly. And that's the basic idea. That is the basic idea. The two are in opposition and can work in opposition. One can strengthen, one can weaken, all depending upon the level of activity in the presynaptic axon. So I think that's really all I wanted to go into. Yeah, that's about that's I don't want to go into anything else. I don't think so. That's the idea. That we can have these persistent activity because of autophosphorylation, but if you have low rates and it occurs a number of times, you can actually reverse that. Okay, that's really all I wanted to talk about. I think is there anything else? No, that's what I don't want to talk about. That's what I wanted to talk. Oh, come on, no, we don't need that. That's what I wanted to talk about up until this exam. So I think all the material is covered, and I know a lot of you are going to have trouble going over this tonight, and that's why I'll be here tomorrow to answer any questions which you might have. So again, I'll ask: does anyone have any questions that they would like to ask or talk about? Or
Yeah, one of the students in the chat, Simon, is asking, "What does our body use LTP for?" I think it's a very loaded question. You'll get into it in later classes, but Dr. Pollack, can you kind of share some applications of LTP?
say this again. What would you use LTP for?
Yeah, yes, that's right.
Don't you learn things when something happens to you? You often remember it. How are you going to remember it if you don't strengthen a synapse? I mean, it seems to be one of the fundamental mechanisms that underlie learning and memory. It's certainly not the only one, but it is certainly one of them. Anyone else have a question I'd like to ask?
I've got a question.
Okay.
Um, I was wondering, like, why would a cell want to like lower the amount of channels it would have, like to,
Why would a cell want to lower what?
like the amount of channels that it has, if it would at all?
Hmm. Well, let me ask you a question. Let's say you didn't have that, and you only had the ability to add on channels. Um, some of your neurons grow until your head would burst. Now, there's got to be a way of weakening synapses as well with low rates of activity, and it's thought, in part, that this plays a role in forgetting. You got to have the opposite to this. You just have to. I can attest to that. Believe me. So one is thought to play. I should mention a few other things too. I mean, we just didn't really have time to get to them yet. But the initial discovery of learning and memory was made um, in a structure called the hippocampus, and it turns out um, these were the first mechanisms. These were the very first mechanisms. These were the very first mechanisms discovered in the hippocampus that correlated with features of learning and forgetting. And they became, for a while, maybe, I don't know, 15, 20 years ago, I mean, you could not pick up a journal without finding numerous articles having to do with long-term potentiation and long-term depression. This was the hottest thing in neuroscience. And these are fundamental properties. That they still are. We know that there are other properties as well, and I'll discuss some of them subsequent to this after the exam. But these are two are very, very, very important. There is one of the things I wanted to show you. We just couldn't do it because of the weather, and we had to eliminate these classes. But what I'm going to go over with you is this absolutely strikingly beautiful paper. It's not perfect. There is no such thing as a perfect paper. The only perfect papers I know that were ever done were done by me. Other than that, there are no perfect papers. But this is fantastic. And what they did is they went on to show that these processes of associative strengthening and weakening really did underlie certain things having to do with learning and memory. And you could eliminate certain memories by doing certain things that weaken synapses, and you could strengthen those memories by inducing long-term potentiation and weaken them and cause forgetting by long-term depression. It's just, they're stunningly. They're stunning articles, just stunning. I wish we had time to go over them before the exam, but we'll go over them next week after the exam.
Dr. Pollack, we have an additional question.
Hmm.
Allison is asking: is there a set number of AMPA receptors already in the brain, or can we produce more of the AMPA
Yeah, you
receptors
can produce
today?
more of them. No, you can produce more of them. Produce, they're easy to produce. They're just proteins. Produce them. That's not doesn't seem to be a problem. Um, it's difficult to grasp how important this was because, as we will see, we're going to talk about a guy by the name of Santiago Ramon Cajal, who was, I think, one of perhaps the greatest of all neuroscientists. We'll talk about him later. And he had predicted something like this. Nobody paid much attention to the ideas of learning and memory that he proposed, but he did propose it. When Hebb came out with this, and I'm going to tell you a lot about Hebb, not more about Hebb, but the school Hebb came from. The school at Montreal, at the Montreal Neurological Institute, when Hebb was there, was probably the greatest school of neuroscience in the world. Reasons we will go into in subsequent lectures. Things were happening there that were beyond belief. And this guy comes up for the first time ever, with a real idea of how neurologically learning and memory could work. And it was a splash. And then when they found it, people were aghast. They were just aghast. And that's how the field works. And that's how it progresses.